Good morning. This is the second part of your mid-year revision or your semester one revision. It includes a group of questions that have been covered before, but whose wording is a bit different. So I thought it would be useful to discuss them with you. The first question says, how is organization of the living systems maintained? Of course, we mentioned before that a living system is a complex organization of molecules or a complex organization of cells or a complex organization of tissues or a complex organization of organs and so on. Meaning that a cell, for example, can be a living system, an organelle can be a living system, an organ can be a living system, a body system can be a living system, the whole human body is a living system, and even a group of living organisms is considered a living system. So how is organization of any living system maintained? Actually, organization means that every living system, starting from the organelle up to the whole living body, has a certain order, and the molecules and processes inside the system have a certain pattern. And this order and that pattern are controlled from the inside of the living system. Let's take, for example, the example of homeostasis. For example, this stability of the pH, stability of temperature, despite all the changes that occur outside the human body, is a good example of how there are internal mechanisms for keeping the internal order and the patterns of that living system, which is the human body. It's also worth noting that if by system we mean, for example, the digestive system or the urinary system or the respiratory system, then we would say a body system. So how is that organization maintained? It is true that it is driven mainly internally, independent of any extrinsic factor, but it also has certain requirements that are related to the external environment. Like, for example, number one, the flow of matter. The flow of matter, for example, you cannot produce energy inside the human body without, for example, getting food, without ingestion of food. And you cannot stay alive without getting rid of, for example, the waste products of metabolism. So you can expel carbon dioxide in exhaled air. You can expel water vapor in exhaled air. You can excrete the surplus of water and salts via the sweat glands and the kidneys. Carbon dioxide can diffuse inside some living cells of plants to photosynthesize. Carbon dioxide can diffuse out of the blood in after cellular respiration, and so on. So there is all the time input of material and output of material. Letter B is the flow of information. Of course, we have studied a lot of examples for flow of information. There is what we can say an internal flow of information, and the internal flow of information is, the internal flow of information is, for example, like the communication between the living cells. For example, we have the signaling molecules like the hormones and other local and remote signaling molecules that occur. So we have the signaling molecules, and the signaling molecules are important for transfer of information or for transduction. So we have, for example, signaling molecules to communicate between the cells, and then we have signaling transduction pathways. We also have, for example, in human beings, the electric impulse transmission in nerves. All these are examples of flow of information inside the human body. And then we have the flow of energy. And as you know, inside the human body, we have two types of processes. We have exergonic or exothermic processes that release energy to drive endergonic or endothermic processes. So this is an example of flow of energy in the body. Number two, how is energy efficiently transferred in a biological pathway? Of course, when we talk about efficient transfer in a biological pathway, this means that we are trying to minimize the losses or minimize the waste, and we are trying to conserve the energy inside the human body. For example, minimizing the waste is by, as we said before, in a biochemical pathway. The biochemical pathway is divided into steps, and maybe if the process is exergonic, we would have a small amount of energy that is released per step, and the body does this because it can handle those small amounts of energy and store them inside the ATP molecules. So this is one way we can minimize the waste of energy by storing that energy into ATP molecules. The second way is by like coupling exergonic and endergonic processes. So for example, in oxidative phosphorylation, the energy of exergonic electron flow is finally stored as ATP. It's true that it is finally stored as ATP, but throughout the process of oxidative phosphorylation, we have had several steps of coupling of exergonic and endergonic. For example, coupling the exergonic electron flow with the endergonic hydrogen pumping. So the second example of minimizing energy losses is the coupling of exergonic and endergonic processes. Okay, and you can give, of course, a multiplicity of examples in that field. Number three, during every energy transfer, entropy increases. And you know that entropy is disorder. As we know that for every energy, of course, transfer is different or differs from transformation. Transformation means that we are converting one type of energy into another type of energy. For example, we can convert electric energy into heat energy. And this is one kind of energy transformation. Of course, heat energy cannot be retrieved. Heat energy is an unusable form of energy. Heat energy cannot be transformed into any other type of energy. This is what we call energy transformation. But even if we are not carrying out one kind of energy transformation, like for example, in the electric iron or in the electric lamp, when, for example, in the electric lamp, this is a good example of how energy transformation can be associated with loss of energy. For example, in the electric bulb, electric energy is essentially converted into light energy, but there is some kind of loss in the form of heat energy, and it is that heat energy that will increase the kinetic energy of the surrounding molecules in the environment, and therefore will result in an increase in entropy or disorder. Okay, number four, how can the structural characteristics of an enzyme make this enzyme very reaction specific? We know that every enzyme has an active domain or an active site, and that active domain is characterized by having a specific shape, and we have to say specific. It has a specific size, and it also has a specific charge. And therefore, if any substrate wants to bind here in the active site of the enzyme and be activated by the enzyme, it has to have complementary characteristics to the enzyme, the substrate, and we'd better say substrate because I've noticed that many of you say reactant. The enzyme active site, shape, size, and charge should be compatible with those of the substrate. And when I say compatible here, I mean complementary, meaning that if the charge on the active site is partially positive, for example, then the charge on the substrate should be partially negative. So this, number 5 asks you to describe an example to your answer in question 4. So for example, amylase is an enzyme that catalyzes the hydrolysis of polysaccharides into disaccharides, and then later on by other enzymes, you can convert the disaccharide into a monosaccharide. Amylase enzyme can catalyze the digestion of the polysaccharide amylose just because amylase has a polar active site and amylose is a polar molecule, and therefore their charge is compatible. So they are not only complementary in shape, complementary in size, or compatible in shape and compatible in size, but they are also compatible in the charge. Number 6, the tertiary shape of an enzyme must be maintained. Justify. Of course, you know that the tertiary shape is essential to have a three-dimensional molecule that has an active domain, and that active domain again has a specific, shape, a specific size, and a specific charge. So if the enzyme loses this specific 3D configuration, then the substrate can no more interact with the active site of the enzyme. So as we said before, the properties of the active site of the enzyme are specific or unique. So unique here means specific. Just like when we said compatible here, compatible means complementary. So you can use either this or that or both if you would like to do so. Number 6, the incorporation of carbon dioxide into a monosaccharide. Of course, this occurs during photosynthesis, and the overall process, as we know, is reduction, does not violate the second law of thermodynamics. What does the second law of thermodynamics state? It states that with every energy transformation, there is some energy that is lost as non-retrievable, non-reusable energy, which is essentially heat energy. So photosynthesis is no exception, because what is the living system of photosynthesis? What is the total living system of photosynthesis? We have the sun that drives the whole process because of the electromagnetic radiation, and then we have the living plants and the cells inside the living plants, and we have the environment outside the plants. So with every energy transfer, with the energy transfer from the sun to the plants, it's true that solar energy is used to excite the electrons, but some of the solar energy is dissipated as heat, and that heat will increase entropy of the environment, meaning that even when the green plant receives solar energy, part of this solar energy is absorbed to achieve the excitation of the electrons inside the pigments of the thylakoids. But meanwhile, there is some energy that is reflected, that is not absorbed by the plant, or even some energy that penetrates the plant and reaches the other side. So all this energy that is reflected and is not or that penetrates the plant and appears from the opposite surface, all this is heat energy that is not used to photosynthesize, but that increases the kinetic energy of the environment. And that dissipated energy, by the way, is important because it helps warmth of the living organisms. Number 8, describe the characteristics of the biochemical pathways. Of course, first of all, they are sequential chemical. Actually, this is not energy. They are sequential chemical reactions, one after the other. And therefore, the fact that they are split into many reactions allows the appearance of several intermediates, and those intermediates can be used laterally in other pathways. It also represents how energy is transformed in living organisms, because when you have a series of chemical reactions, as we said, and at every step you release some energy, then we release energy that can be handled by the cell. As we said before, if this is the initial substrate, the first product, second product, so the product of one chemical reaction can be the substrate of the following chemical reaction. And last thing, when you divide the whole process into several small steps, this allows for more efficient control, because you are more efficient control, because every chemical reaction can be catalyzed by a particular enzyme. This is the first type of control that you can achieve on the reaction. The second type of control is the control of energy. Okay, I'll pause for a second and then resume recording once again. Hello again. We are going to move on to the next slide and see question number 9. Question number 9 says, living cells are in a state of dynamic equilibrium. Of course, we all know the difference between dynamic equilibrium and static equilibrium. An equilibrium is a state in which there is stability and where the movement into the cell is equal to the movement outside the cell. So equilibrium here means constancy or means stability. But is it a like static equilibrium, when everything stays constant and there is no movement? Or is it a state of dynamic equilibrium where everything stays more or less constant? So it is more or less constant, like in homeostasis, for example, but it is a dynamic equilibrium. For example, if we take the example of dynamic pH equilibrium, you're all the time adding acidic ash to your blood by eating proteins, the metabolic products of proteins like amino acids, for example, or uric acid, and you're all the time adding alkaline substances to your blood via your ingestion of fruits and vegetables. But meanwhile, there are alkaline and acidic substances that are excreted either via the kidneys or via the lungs, and there is regulation by the buffer systems of the body. So this is a kind of dynamic pH. You're adding acidic ash, and at the same time, for example, through the kidney, you are removing acidic ash. And the same applies to everything that is kept constant in the blood, even in the flow of material. So there are, for example, like substances that are moving into the cell and substances that are moving outside the cell. For example, you have amino acids that enter the liver cells in order to synthesize proteins to build up, for example, the plasma proteins, but meanwhile, there are amino acids that are, the surplus of amino acids is converted into urea and uric acid. So this is what we mean by dynamic equilibrium. How do cells manage their energy resources? How can you manage your energy resources? Meaning that even if you have a small amount of energy, you're going to keep that a small amount of energy. If you have a lot of energy, then you're going to lose the surplus energy. So this is how we can manage our energy resources. This is one example of managing energy resources. So, but the main process of managing energy resources is by energy coupling, or this is one important example of managing energy resources. So we have the energy releasing processes will be used in energy storing processes. They drive them, they stimulate them to start, and meanwhile, the energy that is released from exothermic processes is stored in endothermic processes. What happens in the mitochondria is reversed in the chloroplast. Justify that statement. Actually, in the mitochondria, the main process that occurs in mitochondria is cellular respiration, and the main process that occurs in chloroplast is photosynthesis. And actually, what occurs in cellular respiration is opposed in photosynthesis. So in mitochondria, for example, we utilize glucose and oxygen in order to form carbon dioxide and water. So the substrates are glucose and oxygen, and the products are carbon dioxide and water. In photosynthesis, it's exactly the reverse. Carbon dioxide and water are the substrates, and glucose is the product. The overall process of cellular respiration is exothermic, but the energy released from cellular respiration will be stored by an endergonic or endothermic process in ATP. The overall process in the chloroplast is endothermic because we are storing the solar light energy in the bonds of the produced glucose molecule. Living systems are self-organizing. As we said before, they do not need external processes to organize them or to keep their pattern or to keep their order. All this arises from internal processes. For example, in the human body, I would call these processes homeostatic processes. Number 13, solar energy is the main energy input for living systems. Of course, yes, because when they hit chloroplasts in autotrophic organisms, the chloroplasts capture the solar energy, and then, as we said, they transform that light energy into energy resources that are usable by all cells. What does this mean? Light energy will be converted into chemical energy. That chemical energy is at first stored in the bonds of the glucose molecule, and then we store them in ATP molecules. Whether glucose or ATP can be used by all living organisms, or like most of them. Number 14, what's the importance of a plasma membrane or of plasma membranes in general? First of all, they create an internal environment that is suitable for the function of the cell. They create their own unique internal environment, and that unique internal environment is not created haphazardly. It is suitable to the requirements of the cell, and therefore they separate the internal environment of the cell from the external environment of the cell. Not only do they create that specific unique internal environment, but they maintain it. For example, if the cell wants potassium to be the main cation extracellularly, and if it wants sodium to be the main cation if it wants potassium to be the main cation intracellularly and sodium to be the main cation extracellularly, then it will do everything possible in order to keep that difference in concentration, and this is mainly achieved by pumping 3 sodium ions outwards and pumping in exchange 2 potassium ions inwards in order to keep that concentration gradient. So usually like creating an internal environment might be easy, but preserving it requires energy. And it wouldn't be able to do this without being selectively permeable. It is the selective permeability that allowed, and we will see how, but here we just say that it is through the selective permeability that of the plasma membrane, of course, that the cell succeeded in creating an internal environment and at the same time succeeded in maintaining it. What's the relationship? How does the selective permeability property help the cell membrane to do this? Number one, by being amphipathic. So we all know that the cell membrane has negatively charged phosphate heads, and then it has a hydrophobic interior made of the 2 layers of lipid tails. So this allows only the non-polar substances to pass, especially if they are small, and prevents the polar substances, especially if they are big. And at the same time, this cell membrane in letter B has proteins, okay? So there could be a question just about the proteins of the cell membrane. These proteins of the cell membrane helps this selectivity of the cell membrane, helps this control function. Selectivity is very related to management and to control, meaning that it would allow certain substances to pass, for example, an ion because of its the presence of its specific channel, or a molecule, for example, glucose, or it would pump substances. So these proteins, and if I ever ask you about the membrane proteins, then you would automatically talk about channels, pumps, and carriers. Channels and carriers are passive, whereas pumps are active. Now, membranes play an important role in compartmentalization, and we all know that compartmentalization is important for specificity, and it is also important for more efficiency from the part of the cell. And we actually have 3 levels of compartmentalization. We have compartmentalization of the whole cell in order to consider the whole cell as a compartment, and this is the role of the cell membrane. So one of the major functions of the cell membrane, as we said before, or the plasma membrane, actually is to separate the internal environment from the external environment. So this is the first level of compartmentalization. And then there are the organelle membranes, and this is a second level of compartmentalization. The membranes of the organelles help create internal compartments, a mitochondrial compartment, a chloroplast compartment, a Golgi apparatus compartment. And then the third level of organization is inside the organelle itself. After having created the mitochondrial compartment, we have further subcompartmentalization through the internal membranes of the organelles. So we have the internal membrane of the mitochondrion that separates it into matrix and outer space. We have the internal membrane of the chloroplast, which is actually the thylakoid membrane, which divides the chloroplast compartment into thylakoids and stroma, and we have the internal folded membranes of Golgi apparatus, which divide the whole compartment of Golgi apparatus into those curved, flattened sacs that are called the cisternae or the cisternae. Now, enzymes, question 7, you see how the content of the answers is easy, but the wording of the question is a bit different and might mislead you, or you might not get the idea of the question. In question number 17, we say that enzymes can be activated by unique, and here we mean specific, and non-unique, non-specific modifications. Of course, the modification here might be a modification in conformation that will finally lead, of course, to a modification in activity, or it might be a direct like modification in activity. The direct modification in activity is through phosphorylation, and it is non-specific, meaning that any enzyme can be phosphorylated by the specific kinase and can be activated, meaning that it is by non-unique here, we mean that phosphorylation is a process that can occur to many enzymes. But there are specific conformational changes that occur uniquely to that enzyme by specific coenzymes, by specific cofactors, or by specific activators. Why are these specific? Because, for example, if we review the function of a coenzyme, what is the main function of the coenzyme? The main function of the coenzyme is to bind with the cofactor actually is to bind with the active site of the enzyme to make it more fitting with the substrate. So it has to have a complementary shape to the shape of the active site. Similarly, cofactors do the same function. The only difference between the two is that coenzymes are organic, coenzymes are large, whereas cofactors are inorganic and cofactors are small. The best example here might be copper or magnesium, and the best example here could be a vitamin B member like NAD, for example, or FAD. Similarly, allosteric activators, they bind with this allosteric site that is different from the active site, but every enzyme has its own allosteric site. That's why we call them unique or specific activators. What are the organelles that are involved in the passage of hereditary information from one generation to another? Of course, in order for the organelle to passing on hereditary information, it should itself contain it should itself contain nucleic acid. So of course we have the nucleus that contains DNA and also contains RNA. But RNA is not essentially a hereditary material as much as it is like a delegate or a deputy of DNA. And we have the mitochondria because inside the mitochondria, you have these multiple small circular DNA molecules that are called the mitochondrial DNA and that are very similar to the prokaryotic DNA, and that support the theory of endosymbiosis. And it is worth noting that you inherit all your mitochondrial DNA from the mom. And of course, if you have a mitochondrial disease, then you must have inherited that disease from your mom. And by the way, most mitochondrial diseases result from recessive mutations, and most mitochondrial diseases are related to energy production, and therefore most mitochondrial diseases are characterized by muscle weakness. Similarly, we have DNA in the chloroplast, and it can be transferred to daughter cells or to the offspring of the plant, but of course, this occurs only in the plants. It is worth saying that the mitochondria are important not only for energy generation and for energy generation and energy storage, but mitochondria are also important in initiation of apoptosis. So mitochondria are important for initiation of apoptosis, and mitochondria are important because they carry the mitochondrial genes that are inherited from the mother. Now, describe the importance of the capsule in the prokaryotes. First of all prokaryotes have cell membranes. All prokaryotes have cell walls, so you have the cell membrane, and then you have the cell wall. But outside the cell wall, you might have a sticky gelatinous capsule, and that sticky gelatinous capsule is not present in all prokaryotes. And then, so the first function is that it helps stickiness of the bacteria to any surface. For example, on your laptop, you might have bacteria that you don't see, but that are sticking to the keyboard. And this is what we call this surface, sticky, moist surface that contains bacteria is considered a biofilm. So if you're ever asked about biofilms, then you have to talk about the capsule of bacteria. In letter B, we say that this gelatinous capsule also helps to resist the defensive mechanisms of the host. For example, you know that some of the white blood cells can act as phagocytes that engulf the bacteria. So those bacteria that have a gelatinous capsule are more protected against phagocytosis than the bacteria that do not have a gelatinous capsule. Therefore, the gelatinous capsule increases the survival of the bacteria, especially in intracellular organisms. What are intracellular organisms? There are bacteria that can live only inside living cells. And of course, they are liable to be destroyed by the biochemistry or the metabolism of the cell and by the immune cells. But when they have a capsule, they are protected. The capsule also protects the cell wall, because as you see, the capsule, the dotted thing here, is outside the cell wall, which is structure number 2 here. So it will protect the cell wall. Protect the cell wall against what? Against antibiotics, for example, because there are many antibiotics and antibacterial substances that act by destructing the cell wall, and protect them against osmotic stress. Osmotic stress is the high pressure, the high turgor pressure that occurs whenever the plant is immersed in a hypotonic solution. The cell wall protects against rupture, but the gelatinous capsule might also imbibe some of the water and prevent part of the water from entering by osmosis as it should enter according to the difference in the water potential. Question number 20 is talking about bioluminescence, and the other word for bioluminescence is living light. And by living light here, we mean that it is light that is produced by living organisms. Essentially, bioluminescence or living light or light produced by living organisms is due to fluorescence. And we know that fluorescence occurs when an electron is excited and jumps from the ground state to the excitation state, or from the lower energy level to a higher energy level according to the quanta of energy it absorbed. And then if that electron is not used, if its energy is not used, for example, by the electron transport chain of photosynthesis, then it will drop again to the ground state, to its original state. But there will always be a difference in energy, and that difference in energy is released as light, as fluorescent light. Okay, so this difference in energy that is released as fluorescent light makes the light energy that is formed of a longer wavelength than the light energy that has been absorbed, because there has been loss of some energy. We have seen bioluminescence or living light in two contexts. The first context is quorum sensing. We know that quorum sensing is a mechanism by which the bacteria can sense that they are many and can respond to being many by emitting light. So we saw that's the relationship between fluorescence and quorum sensing, that the bacteria do not like release fluorescent light or bio light unless they are many. Of course, as you know, this is done through autoinducers. And when the bacteria are many, the autoinducers that diffused outside the bacteria become extremely abundant. And therefore, any new autoinducer, as you know, that will be formed by the bacterium will not be able to diffuse outward. Why? Because there are lots of autoinducers outside that have been secreted by other bacteria. So that autoinducer stays in the bacterium and stimulates the gene for synthesis of luciferase enzyme and luciferin protein. When luciferin, which is the substrate of luciferase, binds with the active side of luciferase, it becomes activated and it emits light. So luciferin is considered a photoprotein because there is a glow that is produced when luciferin reacts with oxygen. So actually, the substrates of luciferase enzyme are luciferin and oxygen. The second bioluminescence has been seen in photosynthesis when the electrons of chlorophyll get excited, and when those excited electrons are supposed to reach the electron transport chain and start the light-dependent reactions. So they're not supposed to return again to the chlorophyll molecule. But when do they return to the chlorophyll molecule? When light is too much intense and many electrons have been excited, exceeding the capacity of the electron transport chain in the light-dependent reaction to accommodate them and accept them, and therefore some of them find that they're not accepted by the electron transport chain, so they return to their original ground state and emit fluorescent light, which is of a longer, again, wavelength than the light that hit the chlorophyll molecule. Now, question 21 is talking about the G protein signaling mechanisms. And it is worth saying that when we discussed, if this is the cell membrane, when we discussed those receptors that are called G protein coupled receptors, then we said that they are essentially receptors. Those are the receptors, but because they are coupled, C stands for coupled, to the G protein, then we call them G protein coupled receptors. Where is the G protein? The G protein is here. So you should be able to differentiate between the G protein that is totally cytosolic and between the receptor that is coupled to the G protein and that we call G protein coupled receptor. That G protein coupled receptor has a part number 1, which is extracellular or extracytosolic, part number 2, which is inside the membrane, and part number 3, which is intracytosolic. When the specific signaling molecule binds with part number 1, there are conformational changes in part number 3. So it is the cytosolic part that changes its shape to activate the G. And when G is activated, it will activate the adenylate cyclase enzyme, to produce ATP. And ATP is considered the secondary messenger because it relays the signal. It allows the signal that stayed outside and that couldn't be able to enter the cell because it is a hydrophilic, for example, signaling molecule, to have a deputy or a delegate inside the cell. So binding of the specific ligand, and it is important to say specific ligand, or the first messenger results in conformational changes in the receptor. In which part of the receptor? In the intracellular domain of the receptor, resulting in activation of the G protein and therefore activation of adenylate cyclase. But how is the G protein activated? As we said before, the G protein is activated as follows. First of all, we know that the G protein consists of three parts. It consists of an alpha part, a beta part, and a gamma part. That's why we say that it is a heterotrimer. A heterotrimer. Why is it a trimer? Because it consists of three parts or three subunits. Why it is a heterotrimer? Because the three subunits are not similar to each other. At the beginning, before activation, these three together form the G protein, not the receptor. At the beginning, they were bonded to each other. Once you see that beta and gamma got separated from alpha, they form together what we call the beta-gamma dimer. You know that G protein has been activated because the three are supposed to be connected to each other. If the beta and gamma separate from each other and form the dimer, then the alpha is alone. And now there will be a very important event that will occur in the alpha. It is physical replacement, meaning that GDP or guanosine diphosphate will leave the domain where it was bonded to the alpha particle, and instead of this, the GTP will stay in the place. Now that GTP, as long as GTP is bonded to the alpha, alpha will stay activated. And therefore, anything that will prevent the binding of GTP to the alpha will result in failure of activation of the G protein. And anything that will prevent dissociation of GTP will keep the alpha in its, activated forever, okay? So we can open a new screen like this and talk about this important process. So here, for example, if we are talking about the alpha subunit of the G protein. So here, there are many questions, or there are always questions about what happens when there is a certain disruption of the normal pathway. So here, disruption of the normal process. Disruption of a normal process can occur either by inhibition of that process or by uncontrolled activation of that process. Uncontrolled activation of this process. Uncontrolled activation of this process. So the alpha unit of the G protein, you might have a question that says, what happens when? What happens when? So the question is, describe what happens when. And in both cases, it would be a disruptive pathway, meaning that we can ask you about, for example, what happens when there is failure of GTP, to bind with alpha. And on the other hand, you might have the opposite exactly. One, of course, they cannot occur at the same time. They cannot occur to the same alpha. It's either this or that. So what can happen if there is a chemical? If we apply to the cell, if we apply a chemical to the cell and that chemical blocks the dissociation. Here it does not block, but it blocks the dissociation of GTP from the alpha particle, okay? This would be an interesting thing to discuss because the processes are opposite. So if there is failure of GTP to bind with alpha, then the G protein will not, the whole G protein will not be activated. Okay? Okay. I'm sorry, I'll pause for a second and then resume again. Hello again. The second consequence or the second option is a chemical block will dissociate, a chemical will block the dissociation of GTP from alpha. Of course, if this is the alpha particle and GTP is already bonded to the alpha particle and GTP stays there, then alpha particle will stay activated. So here, alpha particle will be permanently activated, or at least activated as long as GTP is there, and therefore all the biochemical pathway will continue, or the signal transduction pathway will be continuous. Of course, here when we say that the G protein will not be activated, with all the consequences of the G protein not being activated, meaning that there will not be separation of the beta and gamma dimer, and the whole biochemical pathway would stop. So here we should have continued that the signal transduction pathway would stop. Okay, it wouldn't occur at all. Now, the next question is talking about, Okay. So the next question is talking about the functions of cyclic AMP. Yes, we had a screen in between. I was looking for the screen. Now, we said that activation of the G protein results in activation of adenylate cyclase and the formation of cyclic AMP. What are the consequences of activation of cyclic AMP? What does cyclic AMP do when it is active? Number one, it activates protein kinases. So cyclic AMP, the next step after activation of cyclic AMP is activation of kinases. And kinases are enzymes that activate their own substrates by phosphorylation. Of course, they should activate only their specific substrates, of course, by direct substrate phosphorylation. So the function of the kinase is direct substrate phosphorylation. The second function of cyclic AMP is that, and this we might have not said it in class, it can change the permeability by opening certain channels or closing certain channels. We have just said something about permeability in this kind of signal transduction pathway of the G protein coupled receptor, that the beta and the gamma dimer will go to the cell membrane and might act, the dimer itself might act as an ion channel in the cell membrane. And the third function of cyclic AMP, this is also new, that cyclic AMP can affect the formation of messenger RNA from DNA in the nucleus. How do we call the synthesis of messenger RNA based on the structure of DNA? We call it transcription of messenger RNA. Question 22 is asking about the G proteins, and they are specialized proteins and they are present inside the cell. They can bind, of course, this is recapitulation of what we have said. They can bind to either guanosine diphosphate and therefore be inactive, or to guanosine triphosphate and be active. Okay, the G proteins, the main function of the G protein is to relay the message, as we have said, and activate adenylate cyclase. And when they activate adenylate cyclase, they create that secondary messenger inside the cytosol. And that's why we've converted the primary message or the signal we have converted it into a secondary messenger or a messenger molecule, which is cyclic AMP. Describe the fate of the heterotrimeric G protein when activated. We said before that heterotrimeric means consisting of 3 different units. So upon activation, the first change that occurs upon activation is that we have dissociation of the 3 subunits. The alpha subunit will bind to GTP, and the beta and gamma subunits will form what we call a beta gamma dimer, and that beta gamma dimer will be anchored to the plasma membrane. And as we have just said, it can act as an ion channel to allow the passage of certain ions, or it can diffuse laterally on the cell membrane to interact with other proteins on the cell membrane and do different functions. 24, describe an example of a useful interaction between a lysosome, a vesicle, and a mitochondrion. Of course, if you have a lysosome that is full of hydrolytic enzymes that are capable of intracellular digestion and lysis, if it fuses with a vesicle, and if that vesicle contains macromolecules, it contains big molecules like this, then when they fuse together, this is an interaction. When the 2 membranes fuse together, so the lysosome will open into the vesicle, and the enzymes of the lysosome will enter into the vesicle and digest the macromolecules into small monomers. And then comes the role of the mitochondrion to interact with the already interacted organelles. And you know that the mitochondria can do such type of membranous interaction when we said, for example, that 2 mitochondria can fuse together and exchange their DNA. This is another type of interaction. What's the mitochondria benefiting from this interaction? Getting the monomers that are important for the function of the mitochondria. The monomers might be used in building enzymes, in building DNA, whatever kind of macromolecule that, so the mitochondrion takes the monomer that results from digestion and converts it back into its own macromolecule. Would it be an enzyme or a coenzyme or whatever kind of molecule or a DNA molecule, for example. 25, describe the structure of the cell wall. The cell wall, of course, is made of complex carbohydrates. And of course, we know that it is mainly made of polysaccharides. So the cell wall is essentially made of polysaccharides. And these polysaccharides can be cellulose in plant cells, or can be chitin in fungal cells, or can be peptidoglycans. And by peptidoglycans, as the name indicates, it's protein plus carbohydrate, like the cell walls in prokaryotes or bacteria. And it is worth mentioning that the polysaccharide molecules are macromolecules. They interlace together. For example, if we take the example of cellulose in plant cell walls, and therefore we have like very wide spaces in between the cellulose molecules, and that what makes cellulose or the cell wall freely permeable to water and salts, of course, down concentration gradient. So it is just like a mechanical process of filtration. So the cell wall is freely permeable to water and salts. Okay. Now, 26, the plant cell wall acts as a structural barrier and a permeable barrier. A structural barrier because it gives structural support to the cells. So it protects the cells against all kinds of damage, chemical damage, physical damage. And as we have seen that the cell wall protects the plant against rupture when the plant cell wall becomes turgid. So number one, it protects the cell. Number two, it maintains cell shape. Number three, it prevents cell rupture when the internal water increases. You know that when the plant sap vacuole is turgid and there is turgor pressure, and the turgor pressure is pushing the cell membrane outside, this might cause rupture of the cell membrane if it were an animal cell. But because it's not an animal cell and it is a plant cell, so rupture does not occur. Furthermore, it gives physiological support to the plant. Turgidity gives physiological support to the plant. What do we mean by physiological support? We mean that the plant, by being full of water and by being turgid and not flaccid, this allows the stem to stay upright, to stay perpendicular to the surface of the earth, and allows the surface of the leaves to stay horizontal. And concerning the second part of the question, which is being a permeable barrier, we have seen that the presence of spaces between the interlacing cellulose molecules will allow the cell wall to be freely permeable to water and salts. But down concentration gradient from an area of higher concentration to an area of lower concentration. Similarly, in the cell wall, we have seen before that we have pits, and those pits are areas of the cell wall where we have only primary cell wall and not secondary cell wall. So those pits are thinner than the rest of the parts of the cell wall, and they allow lateral transfer of water between the plant cells. Okay, and now we move on to question number 27, that is talking about auto-induced. So the question says, how can bacteria feel when they are on their own, and how can they feel when they are many? Because you know that for certain functions like bioluminescence or secretion or production of living light, the bacteria do not want to waste their energy. Each bacterium doesn't want to produce a small amount of light that would be inefficient, and therefore they wait until they are many. They wait until this bioluminescence would be efficient because they are abundant, the population is dense, and all the light would accumulate and synergize in order to give strong light. So how do they know this? They know this by secretion of autoinducers through a process of quorum sensing. And whenever you're asked about quorum sensing, you have to say that quorum sensing is a mechanism where the bacteria not only recognize that or feel that they are many, but they respond. So they feel that they are many, or they detect that they are many, and then they respond by secreting biolight. So it's always like this. Whenever you're asked about quorum sensing, you have to mention the two steps: detection, and then after having detected, we should have a response to this. Okay? So first of all, as we said before, they secrete autoinducers. Every bacterium secretes autoinducers. These autoinducers diffuse to the outside of the bacterial cell. And when the bacteria are many and they all secrete autoinducers, then the autoinducers will accumulate inside the cell, in the external environment. When they accumulate inside the external environment, there is no more diffusion gradient for them to pass outwards, so they stay inside the bacteria and they turn on or off certain genes according to the requirements of the bacteria. So again, what is quorum sensing? Quorum sensing is not only the ability to detect bacterial population density, but it is also the ability to respond to high population densities. So how do they respond to high population densities? We don't say by turning on or by turning off certain genes, but by regulation of genes. And regulation of genes just means turning on and turning off, but according to the requirements of the cell, according to the context, according to the situation. So autoinducers may up-regulating the genes that produce them, their own genes, we mean by their own genes, the genes that produce them. And therefore, they say, oh, we are dense now, it's high time to produce BioLite, so let's increase ourselves because we are the autoinducers, we are the ones that will turn on the genes for luciferase enzyme and luciferin protein. So we the autoinducers, also must be numerous. So they turn on their own genes. So the gene has produced an autoinducer, and the autoinducer turns on again its own gene. So it is a kind of positive feedback mechanism. Another thing is that they turn on the gene for luciferase and luciferin. Luciferase is a protein, so it needs a gene to be formed. Luciferase enzyme. But luciferase has, like in its active side, it has two domains. A domain for oxygen and a domain for luciferin protein. When activated, luciferin will react with oxygen. And when it reacts with oxygen and gets oxidized and loses electrons, some of these electrons return to their original ground state and secrete or result in fluorescence. They emit light that is of lower energy. They emit light that is of lower energy because it is of a longer wavelength. And therefore, as we said before, this is why luciferin is considered a photoprotein, just like chlorophyll is considered a photoprotein. So if we open a screen, so we would like write photoproteins. What are photoproteins? Photoproteins are well, first of all, what are photoproteins? They are proteins that show fluorescence. They are proteins that show fluorescence, or they are proteins that emit. They are proteins that emit BioLite. When do they do this? They do this when they are oxidized, meaning that when they lose electrons, and then some of these excited electrons return to their ground state, and the difference in energy makes them emit light. The difference in energy makes them emit light that is of lower energy and therefore of a longer wavelength. The best examples for the photoproteins are chlorophyll, of course, in plant cells, okay, and luciferin protein in bacteria. Unfortunately, we don't, or fortunately, we don't have bioluminescence in our human body. Now, 29, what is the critical mass of their autoinducers? There is a certain concentration of autoinducers that when reached, the autoinducers do not diffuse to the outside and stimulate their own genes. So what is the critical mass? Whenever we talk about critical, then we are talking about a certain limit. So when their extracellular concentration increases, if my extracellular concentration, if for example, we have 4 molecules only of the autoinducer inside, and outside we have 6 molecules, then Now we have a higher concentration outside. So in order for a new molecule of an autoinducer to be transmitted, you will require energy. You will require an active transport mechanism, and that's what we call energetically unfavorable. Energetically unfavorable meaning that if we try to move forcibly these autoinducer molecules, they used to pass by simple diffusion. Yani at the beginning, when autoinducers are released, they are many inside, they are few outside, so they pass by simple diffusion to the outside. But as the number of bacteria increases, and every bacterium has its own input, so the environment is full of autoinducers, so the bacterium finds that it would be energetically unfavorable to pump them out. It would result in a lot of energy, and therefore they stay inside, and their intracellular concentration reaches at that time the critical mass, and they start regulation of gene expression. Now, some membrane receptors have enzymatic activity. Of course, we have seen two major examples of membrane proteins that have enzymatic activity, and that will be added to the functions of the membrane protein. So let's first open a new screen. Okay, so here, if we divide the functions of the membrane proteins, and I think we did this before. So the membrane proteins are many. We can have like transport proteins, okay, transport proteins. And the transport proteins can be like channels, or they can channels, of course, for ions, and then we have the carriers for the molecules, and then we have the active transporters, and the active transporters might be single transporters or co-transporters, or and the co-transporters may be symporters or antiporters. But we also have membrane proteins for recognition, and for identification, like, for example, the glycoproteins that are present on the surface membrane. We also have receptor proteins, like, for example, the G protein coupled receptors and the tyrosine kinase receptors, but we also have membrane proteins that have enzyme action, like the tyrosine kinase receptors. They are not tyrosine kinases, they are tyrosine kinase receptors, because tyrosine kinase are the enzymes that are bonded to the intracellular domain, but the whole receptor is called a tyrosine kinase receptor, and the acronym is TKR. And we also have the adenylate cyclase enzyme, because after activation, it is on the cell membrane. So some membrane receptors have enzymatic activity, and these enzymes will have a membrane part and an extracellular part. And that extracellular part is concerned if this is the membrane, and those are the tyrosine kinases, for example. So this extracellular part will have the domain that will bind with the specific ligand. But here we're talking now about that intracytoplasmic part. It is full of tyrosine kinases. Why do we call them tyrosine kinases? Because here we have tyrosine amino acids that will be phosphorylated tyrosine amino acids. So the enzymatic part of the receptor is inside the cytosol. So they are called tyrosine kinase receptors because they are receptors whose cytoplasmic part contains enzymes. An activated tyrosine kinase can result in multiple cellular responses. Why can it result in multiple cellular responses? Because its intracytoplasmic domain can interact with several relay proteins. As you know, when the tyrosine receptor is activated, it forms a dimer. And after having formed a dimer and after having phosphorylated all the tyrosine residues, there are multiple relay proteins that are activated and that bind to the intracytoplasmic part. And these relay proteins can be up to 10, and every relay protein, when activated, can activate a specific transduction pathway, and that's why they can activate lots of signal transduction pathways. What are the characteristics of signal transduction pathways? First of all, they involve a cascade of molecular interactions. And by cascade, we don't only mean that there is a series. So the term cascade includes or inspires the meaning of a series of chemical reactions one after the other, but it also gives the indication of amplification, like a water cascade, okay? So here we have a cascade of molecular interactions, and by molecular interactions, we simply mean chemical reactions. The second thing is that they're capable of amplifying the appropriate responses that is our target. We are doing this signal transduction pathway and activating it to get a particular cellular response. That cellular response might be cell growth, just growth by a growth factor, or it might be to trigger cell division, mitosis or meiosis, or it might be cell secretion, or it might be gene expression. And when you say gene expression, you're saying a lot, because gene expression means translating the gene into a protein, and that protein might be a hormone, it might be an enzyme. So when we say gene expression, we're actually like talking about all the possible functions of the cell. The third characteristic of a signal transduction pathway, that a signal transduction pathway starts by binding of the first messenger to the receptor, and then after binding of the first messenger to the receptor, we have a series of molecular interactions inside the cytosol because of activation of the intracellular domain, and finally we get a cellular response. So actually we are like relaying the message from the first messenger that unfortunately in most of the time cannot enter the cell because of being hydrophilic. We are relaying the message. What this first messenger or this ligand, when it first came and bonded itself to the receptor, it had a target. That target was to produce a cellular response, but it found out that it was impossible to enter the cell. So through the signal transduction pathway, it could deliver, or it was capable of delivering the message and creating a cellular response inside the cell. How can epinephrine produce different cellular responses? We know that epinephrine in the heart, for example, increases the heart rate. Epinephrine in the skin causes vasoconstriction of the blood vessels of the skin. It increases the blood pressure, and so on. So it has different, in the liver, for example, in the liver, adrenaline, for example, increases glycogenolysis, meaning the breakdown of glycogen. So how could different cells produce different responses if they have the same epinephric receptor or adrenergic receptor? So how can this occur? Because different cells have different receptors to bind with adrenaline. But do you think those different adrenergic receptors would differ in their extracellular domain that binds with epinephrine? Of course not, because if this is the cell membrane and this is the extracellular domain, however different the adrenergic receptor, however different the adrenergic receptor is, the external cytoplasmic domain should be the same because it should be complementary to the shape of adrenaline or epinephrine. So if we say that different cells have different adrenergic receptors, so it will be only in their intracytosolic domain. But how can this intracytoplasmic domain be different if activation of the intracytoplasmic domain always produces cyclic AMP? Yes, it's true that it always produces the same cyclic AMP, but the difference lies in the type of kinase that will be activated by cyclic AMP. Different kinases will be activated by the same cyclic AMP, and each kinase has its specific substrate, and therefore it is activating a different chemical reaction resulting in different cellular responses. Question 34 is talking about amplification of the signal in G protein coupled receptors and activation of the signal in tyrosine kinases. Activation of the signal in G protein coupled receptors is through formation of several cyclic AMP. So you start by one signaling molecule. You start by one signaling molecule, meaning that you start by one ligand, but the binding of that ligand with the receptor produces, for example, 10 to the power 4 cyclic AMP molecules, and every cyclic AMP will activate a kinase. So this is the first amplification. The second amplification is here. So the first amplification is through the formation of a huge number of cyclic AMP from one signaling molecule. The second amplification is here through repeated, each of these 10,000 cyclic AMP molecules will activate a certain kinase repeatedly. And this is how amplification occurs. For the tyrosine kinases, we have just mentioned it. The amplification is through the multiple relay proteins that are bonded to the intracytoplasmic part of the tyrosine kinase. What's the effect of irreversible binding of the GDP to the G protein? As we said before, GDP makes you have to, we said an opposite context, meaning that once you see the question, what's the effect of irreversible binding, you have to understand and focus. What are we talking about? Are we talking about irreversible binding of GDP, or are we talking about irreversible binding of GTP? Because it makes a lot of difference, or a huge difference, because GDP makes the G protein inactive. So if there is irreversible binding, then there would be blocking of the activation of G protein, impossible binding of GTP. Therefore, the G protein will stay inactive and the signal transduction pathway will stop. Describe how water can enter the cell. Of course, you know that water essentially passes through cell membranes by a process of osmosis, and osmosis is considered, by a process of osmosis, and osmosis is considered simple diffusion because the water molecules do not need any help, do not need any assistance. They pass across the cell membrane from an area where they are present in higher concentration to an area where they are present in lower concentration across a selectively permeable membrane. But if you want rapid osmosis, if you want large volumes of water to pass in a small period of time down concentration gradient again, then you should use the aquaporins, and therefore the aquaporins are passive channels for water. So they are passive water channels, and this is a kind of facilitated diffusion. Aquaporins are present in all cells, all living cells, but they are most abundant in the renal tubules and in the collecting ducts. How do ion channels help transport of ions? We know that ion channels are integral membrane proteins and that they have lumens or cavities from inside. We know that they are hydrophobic from outside, and on the other hand, they have a hydrophilic interior. So you must say that they have a hydrophobic outside. Why do they have a hydrophobic outside? In order to interact with the hydrophobic lipid tails, and they have a hydrophilic inside to interact with the charged or the charged ions, yes, that pass inside. What are the cellular structures that are common to all living organisms? Of course, they like this question because when we find out that there are cellular structures that are common to all living organisms. So this is important for evolution, and in order to prove evolution and to prove, as some scientists say, to prove common ancestry of all living organisms. So we have the plasma membranes, we have the ribosomes, and don't say the nucleus, please, because not all organisms are nucleated, but all organisms have some kind of nucleic acid, whether DNA alone or RNA or both DNA and RNA. In slide 39, we're talking about the characteristics of the cancer cells. Cancer cells are characterized by having rapid, uncontrolled cell division by mitosis. However, they do not show contact inhibition because, of course, if they divide rapidly in an uncontrollable way, they would be too crowded. And normally, even in the human body, when the cells are too crowded, they stop. There is a certain regulatory mechanism that stops their cell division. So despite being too crowded, they do not show contact inhibition. Like, why do they rapidly divide? Because they have an abnormal mutated DNA. So an abnormal mutated DNA is the cause, and the rapid uncontrolled division is the effect. Why have we mentioned the effect? Because before the cause, because this is the most striking aspect of cancer cells, that they divide rapidly and uncontrollably. This is due to an abnormal mutated DNA. But this looks weird because if you have a cell that has abnormal and mutated DNA, it should undergo apoptosis. But again, however, they do not undergo apoptosis. They do not follow the rules of apoptosis, and they do not show anchorage dependence. This is the last point. What is anchorage dependent? Normal cells require a basement membrane to rest on in order to be capable of dividing. And therefore, for example, normal cells cannot cannot divide in fluid. They cannot divide in blood, for example. They cannot divide in lymph. But cancer cells do not need all this. They might continue dividing while they are being transported via blood or lymph to other areas, and this is meaning that they can divide even when they are metastasizing. I don't say that this is the cause of metastasis, but they continue dividing while they are metastasizing. And metastasizing is the sixth property. Metastasizing is the sixth property of cancer cells. Why are they capable of metastasizing? Because, for example, I'll erase the ink on the... Why can they metastasize? Because if you have a benign tumor, that the cells inside the benign tumor cannot bypass their capsule, meaning that they have a capsule and they cannot penetrate it. But if you have cancer cells at the beginning, you first of all, we said that we can have a mass of cell without a membrane, but even if there is a capsule around, they will secrete enzymes. They will be able to penetrate that cell membrane and metastasize to distant areas. Now, how is water potential important for plant life? Of course, you know that water potential is the ability of the water molecules to pass from an area where we have a high water potential to an area where we have a low water potential. And therefore, when plant cells are put in an environment that is well irrigated, that is well hydrated and contains enough water, then this would help osmosis from the outside environment to the inside of the cell, from the hypotonic environment that has a higher water potential to the inside of the cell that is hypertonic because of the presence of salts and other solute particles. So first of all, water potential is important for absorption of water by osmosis. And when is absorption of water by osmosis important? Yes, first of all, because water is required for the biochemical processes, in the cell, for example, photosynthesis, but water is also important to give turgidity and support to the plant cell. What's the effect of depriving rapidly dividing cells in culture of, so here it's of phosphate or nitrogen, of nitrogen. Of course, you know that when a plant cell is growing, then it needs all the macromolecules. It needs to have proteins for enzymes, hormones, and organelles. It needs to have carbohydrates for certain structures in the cell membrane. It needs to have fats for the cell membrane, and all these are macromolecules. So it needs carbon, it needs hydrogen, it needs oxygen. But if you're talking about proteins or DNA, that it will also need nitrogen. And if you're talking about DNA, then it will also need phosphorus. It might also need sulfur for the protein. So for DNA, it requires nitrogen and phosphorus. For proteins, it requires nitrogen and sulfur. So if you deprive a growing cell or a group of growing cells from nitrogen and phosphorus, then they will not be capable of forming DNA, and therefore of duplicating DNA, and cell division will stop. Infusion of the secretory vesicles with the plasma membranes. Of course, this is a positive membrane balance. For example, if you have the cisternae of Golgi apparatus, and then we have a secretory vesicle that is released from the Golgi apparatus, that vesicle will migrate until it reaches the cell membrane, and then it will rupture and release its secretion to the external environment. That part is vesicle membrane. That part will be added to the plasma membranes. This results in fusion. How can they fuse together? They have different origins. This one is the plasma membrane, and that one is the vesicle membrane. How can they fuse with each other? Because both of them both are formed of a double layer of phospholipids. If those phosphate heads are the ones for the plasma membrane, and those dotted heads are the ones for the vesicle, so simply what would happen is that phosphate will interact with phosphate. Hydrophilic will interact with hydrophilic, and hydrophobic interior will interact with hydrophobic interior. And this is how any two membranes from any two different sources can fuse with each other. Hydrophilic interacts with hydrophilic, hydrophobic interacts with hydrophobic. And now here we come to a very important question that says that a typical animal cell assumes a normal volume when placed in a solution that has a total solute molarity of 0.3. Deduce the molarity of the cell. Of course, we cannot say SAP here, because, no, actually, I'll cross it out. We cannot say sap here because this is an animal cell, but we can say, for example, cytosol. We can say cytosol. So what does the question, cytosol, what does the question actually say? I'll open a new screen and then move back to the answer. So here, the question says that if you have an animal cell, if you have an animal cell, and that animal cell is placed in a solution where we have several solutes, but the overall molarity of those solutes in the solution is 0.3 molar, okay? Don't forget that molarity is C, meaning that when we talk about the formula of the solute potential, we say that it is minus I C R T. So here is the molarity. And the question tells you that when such an animal cell is immersed for a certain period of time in a solution whose total solute molarity is 0.3, then it stays as it is, meaning that there is no volume change. It does not swell. It does not decrease in volume. No volume change, meaning what? Meaning that there is no swelling and no bursting, but at the same time, it does not lose water, meaning that there is no crenation, there is no shrinking, no crenation, no shrinking, okay? And meanwhile, there are no mass changes like the experiment that you did in the lab. What does this mean? This simply means that the animal cell is in dynamic equilibrium with the outside solution. This means that the cytosol inside the animal cell and the outside solution are isotonic. And when we say isotonic, we mean that they have the same solute concentration. So they are both isotonic here because there is no pressure applied. The pressure potential can be considered zero. But the two solutions are isotonic. What does this? We said before that when we say isotonic, we are talking about the solute potential. So what does this mean? This means that you can take it for granted that an animal cell, that the cytosol of an animal cell has a molarity of 0.3 molar. But there is something to add, is that in order to consider the molarity of the animal cell as being 0.3 molar, it should be, because we told you that there are several solutes in the cell, all the solutes in the solution should be non-ionizable, non-ionizable. This is an important restriction. Why? Because if they are non-ionizable, so I equals 1. So you cannot, for example, get a solution of sodium chloride and say that you cannot say, for example, if you have an animal cell here, you cannot say that if you put it in a solution of 0.3 molar sodium chloride, it would be isotonic. Absolutely wrong. Because if the molarity is the same, yes, it is the same, but the solute potential will be -2 multiplied by the molarity, and then we will have the same R and the same T. So here, you're multiplying by 2. So definitely, if you have the molarity of an animal cell, 0.3 molar of a non-ionizable substance, if you put it in 0.3 molar sodium chloride, it has a greater solute potential, so it is hypertonic, so it will absorb water from the cell. So again, a very important restriction is the one that is written here, non-ionizable solution, okay? Non-ionizable solute. This is concerning 43. Concerning 44, what is a 0.9% sodium chloride solution? Why are we that concerned about a solution of 0.9% sodium chloride solution? Because this is the solution we use for intravenous infusion. Therefore, it is a solution that is isotonic to plasma. Okay, so a 0.9% sodium chloride solution is definitely isotonic to plasma, and therefore definitely isotonic to the contents of an RBC or the contents of an animal cell. But here it is very important to ask yourself one important question. What does 0.9% represent? It is a molarity or is it concentration? No, it is a concentration, meaning that it's not a molar concentration. It is the regular concentration, meaning that when we say 0.9%, of course, it can be represented as a molar concentration, and this is what we're going to see now. But 0.9% sodium chloride solution simply means that we have 0.9 grams of sodium chloride per liter of water, And notice that I wrote sodium chloride in its ionized form in order to show you that I here should be 2. But is it a molar concentration or a concentration? No, it is a concentration. But what about the molar concentration? When we dissolve 0.9 grams, when you dissolve 0.9 grams of sodium chloride in 1 liter, these 0.9 grams represent 1/54 of 1,000 of over 1,000 of the molecular weight of sodium chloride. And therefore, those 0.9 grams that are dissolved in the liter represent 154 milliosmol. If you don't write milli and you divide by, then you have to divide by 1,000. And therefore, the molarity of that solution is 0.154 or just 0.15. N is the number of moles in the per liter. So the molarity of 0.9% sodium chloride is 0.15 molar. Does it make any sense? Yes, it does, because we have just said that sodium chloride is isotonic. So this simply means what? This simply means that this 0.15 molar solution has the same solute potential, as we said, of the 0.3 molar sucrose. And we will prove this again. Why is a 0.9% sodium chloride solution isotonic to plasma, isotonic to animal cells? Isotonic, we can say that it is isotonic to animal cells. We can say that it is isotonic to plasma, because if you say that it is isotonic to animal cells and plasma, then it is definitely isotonic to a 0.3 molar sucrose. Why have we selected sucrose? Because sucrose, the I of sucrose, or the dissociation constant of sucrose, is 1, because it is a non-ionizable particle. So the sucrose molecule is considered 1 particle because it does not split. But the sodium chloride molecule is considered 2 particles. Now let's compare the solute potential. Let's compare the solute potential of a 0.54 molar solution of sodium chloride to the solute potential of a 0.3 molar solution of sucrose. Why are we comparing the solute potentials? Because it is the solute that determines tonicity, and tonicity will determine the direction of water flow. And we will see now how they are interrelated. Now, if you calculate the solute potential for sodium chloride minus ICRT, and here the solute potential for the sucrose solution, of course, if they are under the same conditions, then the constant will cancel the constant and the temperature will cancel the temperature. C will be substituted for by 0.3, that is given in the question. And C here will be substituted for by 0.15, that is also given in the question. And then what will make the difference is the ionization constant. If you multiply 2 by 0.15 and you multiply 1 by 0.3, then on both sides you will have 0.3 here and you will have 0.3 there. So 1-to-1 ratio. Okay, 1-to-1 ratio. So you can say that a 0.154 solution, we can like open a screen and have certain important conclusions. Okay, so we can say that a 0.15 molar solution, a 0.15 molar solution of sodium chloride and a 0.3 molar solution of sucrose or glucose or any non-ionizable substance or any non-ionizable substance, and a 0.9%, 0.9%, يعني 0.9 grams per liter. This is a concentration, not a molar concentration, solution of sodium chloride, which is of particular clinical importance, as I said, because of the infusions, and the plasma and the cytosol inside the cells are all isotonic, are all isotonic. This is extremely important. And now we move on to the next question, and we have here a graph that shows the relation. This is very similar to the one you had on the quiz that shows you the relationship between time in minutes and product concentration in grams. Of course, here we are talking, since you see the plateau, then this is enzyme catalyzed. Since you see the plateau, then this is enzyme catalyzed. But why? Because if you if you have a chemical reaction without an enzyme, okay, so as time passes, you will have a product that will be formed at a slow rate, but it will be steadily formed. But since you have a plateau, then there has been some kind of saturation, and therefore there is an enzyme. So the first conclusion, when you see a plateau, that this is an enzyme catalyzed reaction. The second conclusion that you have to deduce is that we do not know the conditions of the reaction, but since you have a plateau, then you have either you have a limited enzyme or you have a limited substrate, and we will see the difference between them. The first question says, of course, as I said before, whenever you have to draw a graph, you have to be very neat. So first of all, you have to be very neat. This is the first condition. The second condition is that you should have a title for the graph, and then you should label, meaning that you should write here the time, and you should write here the product concentration. This is not the title, by the way. This is labeling of the y-axis. And then you should write the units, minutes, for example, here, and grams there. And then you should have a uniform scale. A uniform scale. And then you should have correct plotting. Let's write them on a separate screen. Let me first erase the ink, okay? And then open a new screen, and how to get a good graph score, okay? Number one, you should have a proper title. Number two, you should label the axes. Number three, you shouldn't forget to write the units. Number four, you should have a uniform scale. on both axes. On both axes. And uniform means uniform, meaning that you have to start by 0, 0, and start the uniform scale from here, okay? And then number 5, proper type of graph. For example, if you have discrete units, you cannot use a line graph. So it might be a line graph, or it might be a bar graph, or it might be a double bar graph, or it might be like just dots, for example, that are not linked together. And then number 6, proper and correct plotting. Okay? The values themselves should be clear. Now, what is the importance here of the intermediate points on the time axis? Of course, to have a more accurate representation, and therefore to have a more accurate interpretation. A more accurate, a more accurate representation, and therefore a more accurate interpretation. What's the relationship between x and y from 1 to 3? Of course, you have a direct relationship. As x increases, y increases. And of course, you know that the change in y over the change in time is the rate of product. So on the x-axis, we don't have the rate. On the y-axis, we don't have the rate. We have the concentration of the product, but when you divide y over x, you get the rate. Another important thing you have to know is that whenever you have an area of the graph where you have a straight line like this, then the rate is constant. It's not 0. The rate is constant. So here, the rate is constant, but since we are increasing, then it is not 0. In the plateau, since you have a straight line, then in the plateau, the rate is also constant, but the rate here is 0. The rate is constant and is equal to 0. Why? Because at any time, so for example, here, if we have at x1, at x2, at x3, you have the same y, so delta y equals 0, okay? In the previous graph, why did you represent by a line graph? Because, of course, the x values are continuous. What is the average rate of product formation during the first 3 minutes? When I say during the first 3 minutes, meaning that during the 3 minutes from 0 to 3, okay? So delta x would be 3 minus 0, and delta y, here is the corresponding value for 3, delta y would be 40 minus 0, okay? So it is 40 over 3, and since it is rate, so here we have, actually it is gram per liter per minute. So it is gram per liter dot minute, okay? Justify the plateau. Of course, when we have a plateau here, this means that the concentration of the product is the same. However, time passes. So it just means that, the concentration of the product is the same. يبقى here we have in the plateau, we have same product concentration. What does this mean? This simply means that during that plateau, why are we saying this? We are saying this because of the y. Whenever you see a plateau, look at the y. Again, whenever you see a plateau, look at the y, because the interpretation of the plateau would be totally different. So if your y is the concentration of the product and you have a plateau, then this simply means that from 3 to 6, for example, we have the same product concentration. What does this mean? That there is no more increase in product. Why isn't there any increase in the product? This will depend upon, is it the enzyme that is the limiting factor, or is it the substrate that is the limiting factor, as we will see on the following slides. So plateau means as time passes, the concentration of the product does not change. One of the examples of causing this is when we have a limited amount of the substrate. When you have a limited amount of substrate in excess of the enzyme, I don't mean in excess of the enzyme. I mean in the presence of an excess of the enzyme. So a limited amount of substrate means here that you have a constant amount of substrate, though you have plenty of the enzyme, though you have plenty of the enzyme. So the enzyme is not the limiting factor, but it is the substrate. So at a certain time, for example, here at minute number 3, all the substrate has been converted into product. So however time passes, there is no more substrate that is full. In question 48, what happens to oxygen consumption in the cell if the ETC is blocked? Of course, oxygen is the final electron acceptor, and if the ETC is blocked, meaning that you have a chemical that stops the ETC, then there wouldn't be electron transfer. Electrons would not reach oxygen, meaning that oxygen will not be reduced, because when electrons reach oxygen, actually, if you have one oxygen atom, 2 hydrogen and 2 electrons, you will form a molecule of water. Okay, of course, you can multiply by 2 and have 2 oxygen atoms, meaning an oxygen molecule plus 4 hydrogen, plus 4 protons, plus 4 electrons will give you 2 molecules of water. Anyways, what happens to the oxygen at the end of the electron transfer chain is a process of reduction. So you can say that we have 4 protons plus 4 electrons plus 1 oxygen molecule will give you 2 molecules of water. So this is reduction of oxygen. If oxygen is not reduced, then it would stay in the cell, and the overall result is that there will be diminished consumption of oxygen. Vitamins of the B group are important for enzyme action. Yes, of course, because first of all, for example, we have several dehydrogenase enzymes. First of all, let's start by them being hydrogen carriers. What are the best members of the vitamin B complex that act as hydrogen carriers? We have NAD that carries hydrogen and is reduced into reduced NAD, and we have FAD that is reduced into FAD. So by being hydrogen carriers, they help removal of hydrogen from the substrate. So therefore, they help oxidation of the substrate, for example, as they did in case of glucose, for example. Okay, but how is this related to being coenzymes? Being coenzymes means what? As we said before, that they might bind with the active site to make it more fitting with the substrate. So the first action is that they act as hydrogen carriers. They receive the removed hydrogen, and when they receive the removed hydrogen, then they help oxidation of the substrate. So they themselves get reduced and help oxidation of the substrate. On the other hand, they act as coenzymes for many enzymes, like, for example, pyruvate dehydrogenase. We also have lactate dehydrogenase, but we also have NAD dehydrogenase and FAD dehydrogenase. So they are not only coenzymes they are coenzymes for pyruvate dehydrogenase and lactate dehydrogenase, but they are also coenzymes for NAD dehydrogenase and FAD dehydrogenase. Not only are they like coenzymes, but they are actual substrates. For example, if we have the NAD dehydrogenase here, then NAD is a substrate for the NAD dehydrogenase. It helps oxidation of NAD but by converting it into oxidized NAD. So actually, I'm sorry, I made a mistake here. They are coenzymes, sorry. They are coenzymes for pyruvate dehydrogenase and lactate dehydrogenase, but I'm sorry, I apologize for this mistake. They are substrates for NAD dehydrogenase and FAD dehydrogenase. So again, let's recapitulate because of the mistake that occurred here. First of all, they act as hydrogen carriers because they receive hydrogen atoms. And when they receive hydrogen atoms, they get reduced themselves and they help oxidation of other substrates. Meanwhile, they act as coenzymes for certain dehydrogenases like the pyruvate dehydrogenase and the lactate dehydrogenase. Why are there good coenzymes for these enzymes? Because whether we are talking about the pyruvate dehydrogenase or lactate dehydrogenase, they catalyze removal of hydrogen atoms, and this is dehydrogenation. So they catalyze removal of hydrogen atoms from pyruvate and therefore oxidation of pyruvate. They catalyze oxidation of lactic acid. So there is removal of hydrogen atoms, and these hydrogen atoms will be carried by NAD and FAD. But meanwhile, they are substrates for other dehydrogenases like the NAD dehydrogenase and the FAD dehydrogenase, where NAD is not a hydrogen carrier only, but it comes with its hydrogen, carrying its hydrogen, and it is the NAD dehydrogenase that removes the hydrogen and oxidizes NAD. Why is it important to reoxidize NAD? So that we can unload NAD. So any reduced NAD that results from glycolysis or Krebs cycle has to unload. So let's open a new screen to see NAD dehydrogenase enzyme and how it acts. So here we have, for example, the electron transport chain. And here we have glycolysis, okay, in the cell, in the cytosol. And then we have Krebs cycle. And from glycolysis and the Krebs cycle, we have reduced NAD. Here, what did reduced NAD serve as? It served as a hydrogen carrier, okay? But in that reduced NAD has to unload by giving its protons to the electron transport chain. What helps it to unload is an enzyme that is called the NAD dehydrogenase. That enzyme will catalyze the transfer of the two hydrogen atoms to the electron transfer chain. So it will catalyze transfer of the two hydrogen atoms to the electron transfer chain, and therefore it catalyzes the conversion of reduced NAD into NAD. Is it important? Yes, because this helps unloading of NAD. So you might have a question that tells you what happens when. You might have a question that tells you what happens when. What happens when this NAD dehydrogenase is not functioning? If it's not functioning, then reduced NAD will stay reduced. This is number one. Number two, it will not unload. So if it does not unload to the electron transfer chain, then the electron transfer chain stops. Furthermore, if reduced NAD stays reduced NAD, so as time passes, glycolysis and the Krebs cycle will stop. But what will stop first is the Krebs cycle. Sometimes when reduced NAD stays reduced, okay, there is a very important process that occurs. So we're going to open a new screen, and we're going to open a new screen, okay? Or actually, we're going to erase the ink on the screen and say, what happens, okay, when NAD dehydrogenase stops functioning. For example, it might stop functioning by a mutation. So first thing is that reduced NAD will stay reduced, okay? The first immediate consequence is that ETC stops. And if ETC stops, then ATP production stops, and the cell would eventually die, of course. The second important thing, that Krebs cycle stops, okay? And if Krebs cycle stops, the most immediate event, that pyruvate, unfortunately, I'll not be able to, okay, let's say, okay, and then let me stop sharing and reshare again. Okay, let me stop sharing and reshare the screen again. Okay, and then get the slideshow. Okay, and then stop sharing to make sure we are on the right slide, and then reshare. Oh, it's a complicated process. And of course, the screen disappeared. Did it disappear actually? Yes, it did. Both screens disappeared. Okay. However, I'll be repeating the question again on a new screen. So here we have, if NAD dehydrogenase is not working, so if NAD dehydrogenase stops functioning, if NAD dehydrogenase stops functioning, then reduced NAD will stay reduced, The first direct consequence of this is that the ETC will stop because it does not receive electrons, and therefore ATP formation will not occur. Number two, Krebs cycle will stop. Krebs cycle will stop. And if Krebs cycle stops, the most important consequence is that pyruvate does not enter the mitochondrion, and pyruvate will be shifted to fermentation. Pyruvate will be shifted to fermentation. And then finally, at the end of the day, glycolysis itself would stop and the cell would die. Okay, the next question, the next question is, justify the plateau in each of the following graphs. Here we have on the x-axis, let's first take this one, time in minutes. Let's first take the graph C, or graph C, and graph C has time in minutes on the x-axis and has the product concentration on the y-axis. And here we said that the plateau simply means that there is no more formation of the product, okay? There is no more formation of the product, maybe because all the substrate has been converted into product. This is the cause in graph C. Now, let's see graph B. Graph B says enzyme concentration and the rate of the reaction in grams per minute. I'll stop for a while, please. I'll pause. Now we have to take into consideration here that this graph B says what? Says that as you increase enzyme concentration, say for example, we start here at point A and here at point B. So from A to B, as you increase the enzyme concentration, product formation or the rate of the reaction increases. But after point B, as you increase enzyme concentration, the rate of the reaction stays the same. So here you have a constant rate. You don't have here, you have a constant rate. You don't have a zero rate. Why? Because here what is on the y-axis is the rate of the reaction. So you just say that the rate does not change. This is number one. So the interpretation, if you want to interpret only, then from point A to point B, as you have to look very carefully what you have on the x-axis and what you have on the y-axis. Here, you don't have time on the x-axis. You have enzyme concentration, and on the y-axis, you have the rate of the reaction. So from point A to point B, as you increase enzyme concentration, the rate of the reaction increases. And that sounds logical because as you add more and more of the enzymes, more active sites are available for the substrate, more collisions occur with the substrate. So here you increase the enzyme, then you are increasing the active sites, allowing more substrate to bind, and the rate increases. But after a certain, after point B, the rate stays constant. However, the enzyme concentration increases. Here, the substrate concentration is the limiting factor. So after point B, substrate concentration is limiting, meaning that we cannot increase the rate of the this doesn't mean, by the way, that the product is no more formed. It is formed, but at a constant rate, although we are increasing the enzyme because we are limited by the substrate concentration. Here we have the opposite. If we, for example, we call that point A and that point B. So as you increase the substrate concentration, here we have substrate, the rate of the reaction increases up to a certain point. When you increase the substrate concentration, but you still have a constant rate, so at that point, it is the enzyme concentration that would be the limiting factor. What is the monomer of the kinase enzyme? Kinase enzyme and all enzymes have amino acids as their monomers. What are the results of breakdown of an enzyme? Of course, how do we break down enzymes? We break them down by hydrolysis, and hydrolysis gives amino acids. How do pH and temperature affect enzyme action? As you go away from the optimum pH and temperature, then they would affect the types of bonds. They would affect the types of bonds mainly between the R radicals, and this is very important to say. So breaking bonds or changing bonds, affect here means change the bonds, but it is worth mentioning that they are the bonds between the R radicals because these are the bonds that determine the RD configuration, and therefore they determine the size, and this is very important to mention, the size, the shape, and the charge of the domain of the active site. Okay, what's the relationship between the primary and tertiary structures? Of course, you know that the primary structure is the linear polypeptides. Of course, every linear polypeptide, this is the primary structure, and a linear polypeptide is made of amino acids. And most importantly, every amino acid has a particular R. Are the R's important for the secondary structure? No, they are important for the tertiary structure because they provide regionality to the amino acid. How? Because these R's may be polar or may be non-polar. And if they are polar, they might be acidic, or they might be basic, and therefore we would have different types of interactions in the tertiary structure. Discuss the importance of carbon to life and discuss the importance of water to life. Of course, the importance of carbon to life, first of all, it forms the carbon skeleton in all macromolecules, and that carbon skeleton can be large. It can form different types of bonds. It can form single bonds with other atoms, double bonds, and even triple bonds with even another carbon atom or with different carbon atoms, and therefore it is important for formation of all types of macromolecules in life, and all these bonds will store energy, and that's why, of course, energy or carbon, organic carbon compounds are important for life because they are a source of energy, and that energy can be released through the process of cellular respiration. That energy is stored in the process of photosynthesis, and it is released in the process of cellular respiration. The importance of water to life, of course, water is important as a solvent for many substances. Water is important by being a solvent. It acts as a medium for biochemical reactions, and by being a solvent, it would help transport of certain substances dissolved in water. Water is important to moderate temperatures since it has a specific heat capacity, moderate temperature on Earth, and also important to regulate your body temperature. Regulate your body temperature. Water is also important in the process of hydrolysis, and therefore in the process of digestion, whether you're talking about intracellular or extracellular digestion. Water is also important for turgidity of the plant cells, and therefore for support. We have plenty of benefits of water, of course, and most of these characteristics, or almost all of them, arise from the polar characteristics of water. Here, of course, if you're asked about the water movement will depend on water potential. Water potential depends on solute potential and pressure potential, but here the pressure potential on both sides is the same since they are both exposed to atmospheric pressure. And of course, you know that a solution that is 0.15 sodium chloride is isotonic to a solution, so of 0.3 molar sucrose, then we would say that there is no net movement. Justify your answer to A. Of course, in order to justify, you have to apply that formula ICRT both for sodium chloride and for sucrose, and substitute for C, substitute for I, and you would find that the ratio is 1:1. So the animal cell molarity is 0.3. The animal cell molarity is 0.3. If we are talking about a substance that is I equals 1, or whose I equals 1. Examples of substances whose I equals 1. We have, for example, glucose, we have sucrose, we have amylose, all these substances that are non-ionizable substances. Non-ionizable substances. Whereas if the substance has an I of 2, like sodium chloride, for example, then the molarity would be only 0.50. Of course, I wish that video was a comprehensive, or was useful to you, and I wish the video included a comprehensive revision of all the important points. I also wish that that video could have delivered a message that is the wording of some questions might look a bit weird, but you don't have to panic, because since you have the content, since you understand the main concepts, then you can make use of those important concepts to give correct and relevant answers. Most of the time, you write correct answers. Most of the time, you have all the concepts you need in order to answer the question. But the major problem is that we have to read the question carefully in order to avoid writing irrelevant questions, irrelevant answers. Okay, I hope this video was useful. I know it's lengthy, but it's a kind of comprehensive revision. Thank you so much, and I wish you the best of luck in your semester 1 tests, inshallah. Thank you and bye.